हेलो दोस्तों, आप सभी का स्वागत है हमारे यूट्यूब चैनल पे। आज हम एनसीईआरटी रीडिंग कर रहे हैं। हमारे चैप्टर का नाम है चैप्टर 9 बायोमोलिक्यूल्स। देयर इज़ अ वाइड डाइवर्सिटी इन लिविंग ऑर्गेनिज्म इन आवर बायोस्फीयर। नाउ अ क्वेश्चन दैट अराइज इन आवर माइंड इज, आर ऑल लिविंग ऑर्गेनिज्म मेड अप ऑफ सेम केमिकल्स? That is why element and compounds. You have learned in the chemistry how elemental analysis is performed. If we perform such an analysis on a plant tissue, animal tissue, or a microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, and several others, and their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of earth crust, as an example of the non-living matter, we obtain a similar list. What are the difference between the two lists in absolute terms? No such differences could be made out. All the elements present in the sample of earth crust are also present in the sample of the living tissue. However, a closer examination reveals that the relative abundance of the carbon and hydrogen with respect to other elements with higher in any living organism than in the earth crust. Table 9.1. How to analyze chemical composition? We can continue asking in the same way, what type of organic compound are found in living organisms? How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue, a vegetable or a piece of liver, etc., and grind it in a trichloroacetic acid Cl3COOH using a mortar and a pestle. We obtain a thick slurry. If we were to strain this through a cheesecloth or cotton, we would obtain two fractions. One is called the filtrate, or more technically, the acid soluble pool, and the second, the retenant, or the acid insoluble fraction. Scientists have found thousands of organic compounds in the acid soluble pool. In the higher class, you will learn about how to analyze a living tissue sample and identify a particular organic compound. It will suffice to say here that one extract the compound, then the subject the extract to various separation technique till one has separate a compound from all other compounds. In other words, one isolates and purifies the compound. Analytical techniques, when applied to the compound, give us an idea of a molecular formula and the probable structure of the compound, and all the carbon compounds that we get from living tissue can be called biomolecules. However, living organisms have also got inorganic elements and compound in them. How do we know this? A slightly different but destructive experiment has to be done. One weighs a small amount of a living tissue, say a leaf or liver, and this is called wet weight, and dry it. All the water evaporates, the remaining material gives dry weight. Now, if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form, CO2, water vapor, and are removed. What is remaining is called ash. This ash contains inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid soluble fraction. Therefore, element analysis gives elemental composition of living tissue in the form of hydrogen, oxygen, chlorine, carbon, etc., while analysis for compounds give an idea of the kind of organic, figure 9.1, and inorganic constituent, table 9.2, present in the living tissue. From a chemistry point of view, one can identify functional group like aldehydes, ketone, aromatic compound, etc. But from a biological point of view, we can classify them into amino acid, nucleotide base, fatty acid, etc. Amino acids are organic compounds containing an group, amino group, and the acidic group as substituents on the same carbon. That is why the alpha carbon, hence they are called alpha amino acids. They are substituted methanes. There are four substituent groups occupying the 4 valency position. These are hydrogen, carboxyl group, amino group, and variable group, designated as R group. Based on the nature of R group, there are many amino acids. However, those which occur in protein are only of 20. Types of R group in the proteinaceous amino acids could be a hydrogen. The amino acid is called glycine. a methyl group alanine, hydroxyl methyl serine, etc. 3 of the 20 are shown in figure 9.1. The chemical and physical properties of amino acid are essentially of the amino, carboxyl and R functional group. Based on number of amino and carboxyl group, there are acidic, example glutamic acid, basic, lysine and neutral valine amino acids. Similarly, there are aromatic acids, tyrosine, phenylalanine, tryptophan. A particular property of amino acid is the ionizable nature of NH2 and COOH groups. Hence, in solutions of the different pH, the structure of amino acids changes. B is called deuterionic form. Lipids are generally water insoluble. They could be simple fatty acids. A fatty acid has a carbonyl group attached to an R group. The R group could be a methyl CH3 or ethyl C2H5 or higher number of CH2 group, 1 carbon to 19 carbons. For example, palmitic acid has 16 carbon including carboxyl carbon. Arachidonic acid has 20 carbon atoms including the carboxyl carbon. Fatty acid could be the saturated without double bond or unsaturated with one or more carbon-carbon double bond. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both the glycerol and fatty acids. Here the fatty acids are found esterified with glycerol. They can be then monoglyceride, diglyceride, triglyceride. These are also called the fats and oils based on melting point. Oils have lower melting point. Example gingerly oil, hence remain as oil in winter. Can you identify form of the market? Some of lipids have phosphorus and phosphorylated organic compounds in them. These are phospholipids. They are found in cell membrane. Lecithin is one example. Some tissue, especially the neural tissue, have lipids with more complex structure. Living organisms have a number of carbon compounds in with the adenine, which heterocyclic rings can be found. Some of these are nitrogenous bases: adenine, guanine, cytosine, uracil and thymine. When found attached to a sugar, they are called nucleosides. If a phosphate group is also found esterified to the sugar, they are called nucleotides. Adenosine, guanosine, thymidine, uridine and cytidine are nucleosides. Adenylic acid, thymidyllic acid and guanylic, uridylic and cytidylic are nucleotides. Nucleic acids like DNA and RNA consist of nucleotides only. DNA and RNA function as genetic material. 9.2 Primary and secondary metabolites. Most existing aspect of chemistry deals with isolating thousands of compounds, small and big, from living organisms, determining their structure and if possible synthesizing them. If one were to make a list of biomolecules, such a list would have thousands of organic compounds, including amino acids, sugar, etc. For the reasons that are given in the section 9.10, we can call these biomolecules as metabolites in animal tissue. One notices the presence of all such categories of compounds shown in figure 9.1. These are called primary metabolites. However, when one analyzes plant fungal and microbial cells, one would see thousands of compounds other than these called primary metabolites. Example: alkaloids, flavonoids, rubber, essential oils, antibiotics, color pigment, scents, gums, spices. These are called secondary metabolites. Table 9.3. While primary metabolites have identifiable function and play known roles in normal physiological process, we do not at the moment understand the role or functions of all the secondary metabolites in host organisms. However, many of them are useful to human welfare. Example: rubber, drugs, spices, scents and pigments. Some secondary metabolites have ecological importance. In the lateral chapter and years you will learn more about this. 9.3 Biomacromolecules. There is one feature common to all those compounds found in the acid soluble pool. They have molecular weight ranging from 18 to around 800 Dalton approximately. The acid insoluble fraction has only 4 types of organic compound, that is proteins, nucleic acids, polysaccharides and lipids. These classes of compounds, with the exception of lipid, have molecular weight in the range of the 10,000 Dalton and above. For these very reasons, biomolecules, that is, chemical compounds found in living organisms, are of two types. One, those which have molecular weight less than 1000 Dalton and usually referred to as macromolecules or simply biomolecules, while those which are found in acid insoluble fraction are called macromolecules or biomacromolecules. The molecules in the insoluble fraction, with the exception of lipid, are polymeric substances. Then why do lipids whose molecular weight do not exceed 800 Dalton come under acid insoluble fraction? That is why macromolecular fractions. Lipids are indeed small molecular weight compounds are compounds and are present not only as such but also arranged into structures like cell membrane and other membranes. When we grind a tissue, we are disrupting the cell structure. Cell membrane and other membranes are broken into pieces and form vesicles which are not water soluble. Therefore, these membrane fragments in the form of vesicles get separated along with the acid insoluble pool, and hence in the molecular macromolecular fractions, lipids are not strictly macromolecules. The acid soluble pool represent roughly the cytoplasmic compositions. The macromolecules from cytoplasm and cell and organelles become the acid insoluble fractions. Together they represent the entire chemical composition of living tissue or organisms. In summary, if we represent the chemical composition of living tissue from abundance point of view and arrange them class wise, we observe that water is most abundant chemical in the living organism. Table 9.4. 9.4 Proteins. Proteins are polypeptide. They are liner chains of amino acid linked by peptide bonds as shown in the figure 9.3. Each protein is a polymer of amino acid, as there are 20 types of amino acid. Example: alanine, cysteine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer, repeating n number of times. This information about the amino acid content is important, as later in your nutrition lesson you will learn that certain amino acids are essential for our health and they have to be supplied through our diet. Hence, dietary protein are the source of essential amino acid. Therefore, amino acids can be essential or non-essential. The latter are those which our body can make, while we get essential amino acids through our diet, food. Proteins carry out many functions in living organisms. Some transport nutrients across the cell membrane, some fight infectious organisms, some are hormones, some are enzymes, etc. Table 9.5. Collagen is the most abundant protein in animal world, and ribulose biphosphate carboxylase oxygenase rubisco is the most abundant protein in whole the biosphere. 9.5 Polysaccharide. The acid insoluble pellet also has polysaccharide carbohydrate as another class of macromolecules. Polysaccharides are long chain of the sugar. They are thread, literally a cotton thread containing different monosaccharide as building block. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. Cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of energy. In plant tissue, animals have another variant called glycogen. Inulin is a polymer of fructose. In a polysaccharide chain, say glycogen, a right end of called the reducing end and left end is called the non-reducing end. It has branches as shown in the figure form of a cartoon. Figure 9.2. Starch form helical secondary structures. In fact, starch can hold I2 molecule in the helical portion. The starch I2 is blue in color. Cellulose does not contain complex helices and hence cannot hold I2. Plant cell was are made up of cellulose. Paper made up of plant pulp and cotton fiber is cellulosic. There are more complex polysaccharides in nature. They have as building blocks amino sugar and chemically modified sugars. Example glucosamine, N-acetyl glucosamine, etc. Exoskeletons of arthropods, for example, have a complex polysaccharide called chitin. These complex polysaccharides are mostly homopolymers. 9.6 Nucleic acid. The other type of macromolecule that one would find in the acid insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides. Together with polysaccharides and polypeptides, this comprises the true macromolecular fractions of any living tissue or cell. For nucleic acid, the building block is a nucleotide. A nucleotide has three chemically distinct components. One is a heterocyclic compound, the second is a monosaccharide, and the third a phosphoric acid or phosphate. As you notice in figure 9.1, the heterocyclic compound is nucleic acid are the nitrogenous base named adenine, guanine, uracil, cytosine and thymine. Adenine and guanine are substituted purines while the rest are substituted pyrimidines. The skeletal heterocyclic rings is called as purine and pyrimidine respectively. The sugar found in the polynucleotides is either ribose, a monosaccharide pentose, or 2'-deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid DNA, while which contains ribose is called ribonucleic acid. 9.7 Structure of proteins. Protein as mentioned earlier are heteropolymers containing string.